Hello everyone, welcome to SL Biology. In this lesson, we will discuss the question number 1 to 5 of 2021 GCE A Level Biology paper in English medium. So question 1, which of the following is a non-reducing sugar? Ribose, lactose, maltose, galactose, sucrose. So to find the answer for this question, we first have to know what are reducing sugars and what are non-reducing sugars. When we learn monosaccharides, we learn 3 monosaccharides: glucose, fructose and galactose. When these monosaccharides form bonds with another monosaccharide, they form disaccharides. When a glucose molecule binds with another glucose molecule, it gives maltose. When fructose reacts with a glucose molecule, that gives sucrose, releasing water. When galactose reacts with glucose, it gives lactose, again releasing water. Out of these monosaccharides and disaccharides, except sucrose, all the other sugars are reducing sugars. Sucrose is a non-reducing sugar. So what's the difference between these reducing and non-reducing sugars? A reducing sugar donates electrons to another chemical substance. They have a free aldehyde or a keto group in the molecule. So they can undergo oxidation reduction reactions, reducing the other compound. So they are called reducing sugars. A non-reducing sugar does not have a free aldehyde or keto group, so they cannot donate electrons. We can identify these or separate these reducing and non-reducing sugars using a Fehling's test or a Benedict test. In those reactions, reducing sugars can reduce copper ions, changing the color of the compound. So we can use those tests to identify reducing sugars. So if we look at the structures of these compounds, we will first take the structure of glucose and fructose. Glucose is a 6-carbon compound. In this glucose molecule, we can see this aldehyde functional group. And if we take the structure of fructose, again that is a 6-carbon compound. So this fructose has this C double bond O keto functional group. So because of the presence of these free aldehyde and keto groups, they can act as reducing sugars. Now let's look at the question again. In this question, first answer ribose. Ribose is a pentose sugar that is also a reducing sugar. I will draw the structure here, but you're not supposed to remember these structures. So this pentose sugar, ribose sugar, is a reducing sugar. Then the second one, lactose. Lactose is a disaccharide. So it is the product of the reaction between glucose and galactose. Again, I will draw the structure, but we don't have to remember these. Here, due to the presence of this group, lactose can act as a reducing sugar. Then the third one, maltose. Again, maltose is a disaccharide. Maltose is the product between the reaction of two glucose molecules. That is again a reducing sugar. Fourth one, galactose. I will draw the structure of galactose molecule. That is again a 6-carbon monosaccharide. So this one has this free aldehyde group, CHO group. So it acts as a reducing sugar. Then the last one, sucrose. Sucrose is a disaccharide, the product of the reaction between glucose and fructose. Sucrose is unable to act as a reducing sugar. So the only non-reducing sugar here is sucrose. The answer for this question is sucrose, that is a non-reducing sugar. Then question number 2. Which of the following statements regarding plasma membrane is correct? It is mainly composed of carbohydrates, phospholipids, and proteins. Plasma membrane has phospholipids and proteins as the two major components, but not carbohydrates. Carbohydrates are sometimes found in the plasma membrane. I will quickly draw a diagram of the plasma membrane. So it has a phospholipid bilayer. And proteins. Sometimes we find polysaccharides or carbohydrates attached to these phospholipids or protein molecules. So the two major compounds of the plasma membrane are phospholipids and proteins, not carbohydrates. So number 1 is incorrect. Then second one, phospholipid molecules are movable and provide a fluid nature to the membrane. Yes, these phospholipid molecules are movable, so they provide a fluid nature to this membrane, and the arrangement of these protein molecules give the mosaic nature. So that's why we say this model is a fluid mosaic model. So option answer 2 is correct. Then number 3, peripheral proteins are tightly attached to the outer surface of the membrane. Peripheral proteins are the ones lie on these outer layers of the membrane. Peripheral proteins are the proteins that are loosely bound or temporarily bound to the surface of the membrane. They are not tightly bound. They are not tightly attached to the outer surface of the membrane. So option 3 is also incorrect. Then 4, phospholipid bilayer enables nearby cells to communicate with each other. This communication is done by proteins and also polysaccharide molecules. Proteins embedded in this plasma membrane help to identify the cell and enable the cells to communicate with each other. So they involve in cell recognition. So it is not the phospholipid bilayer. So 4 is incorrect. Then number 5, hydrophobic tails of phospholipids attach to cytoskeletal fibers and help to maintain the shape of the cell. Hydrophobic tail is here in this phospholipid molecule. There are two ends, hydrophilic head and hydrophobic tail. The tail does not like water. They always try to stay in the other side of the membrane. Here this answer says hydrophobic tails of phospholipids attach to cytoskeletal fibers and help to maintain the shape of the cell. The hydrophobic tails are facing towards the inner side of the plasma membrane, so there is no chance for them to attach to cytoskeletal fibers and help to maintain the shape of the cell. So number 5 is incorrect. The correct answer for this question is number 2. Phospholipid molecules are movable and provide a fluid nature to the membrane. Then question number 3, select the correct subcellular component function combination. First one, glyoxysomes, transport of residue materials out of the cell. So glyoxysome is a specialized type of peroxisome. They are just simple cell organelles. They're found in plants. They convert fatty acids to sugar. So this one is incorrect. They don't transport, they're not involved in transport of residue materials out of the cell. So number 1 is incorrect. Then 2, smooth endoplasmic reticulum, production of transport vesicles. There are 2 types of ER. There are 2 types of ER. One type has ribosomes on top of the membrane. This is called RER or rough ER. The other type, they don't have ribosomes on that membrane, so they are smooth. They are called smooth ER or SER. So the second answer, smooth endoplasmic reticulum, production of transport vesicles. Yes, they are they do that. They synthesize lipids like oils, steroids, and phospholipids, and they produce transport vesicles to transport within cells. So they produce vesicles. So second one is correct. Then third one, rough endoplasmic reticulum, metabolism of carbohydrates. No, they don't involve in this metabolism of carbohydrates. The function of rough ER is rough ER has ribosomes on the membrane, so they are involved in things related to proteins. So RER, or rough endoplasmic reticulum, they transport proteins synthesized by those ribosomes, and they synthesize glycoproteins, and also they produce transport vesicles. They are mainly involved in synthesizing and transporting protein related things. They don't do this metabolism of carbohydrates. So 3 is incorrect. Then 4, nucleus synthesis of glycoproteins. That is a function of RER. Glycoproteins, synthesis of glycoproteins is a function of RER, not a function of nucleus. Nucleus is the cell organelle that controls all the cellular activities. It synthesizes DNA and RNA, rRNA. So they're not involved in this glycoprotein synthesis. So option 4 is incorrect. Then number 5, peroxisomes photorespiration. Peroxisomes are again single membrane simple structures. They are found in both plants and animals. They have enzymes that catalyze the breakdown of H2O2, hydrogen peroxide. So the function of peroxisomes is detoxification of peroxicides, but in plants they do photorespiration. So number 5 is also correct. So this question has two answers. Both 2 and 5 are correct. Question number 4. 4 events of meiosis are given below. A. Centrosomes move towards opposite poles, forming spindle. Formation of synaptonymal complex. Pairs of homologous chromosomes arranged on metaphase plate. Crossing over of chromatids. Which one of the following is the correct sequence of occurrence of above events? So, to do this, let's see in which phase of meiosis these events occur. First one, A. Centrosomes move towards opposite poles, forming spindle. Centrosome is the complex of centromeres. So they move towards the two opposite poles in prophase 1. In meiosis, we can see two divisions, meiosis 1 and meiosis 2. So here we are talking about meiosis 1 because it talks about crossing over, synaptonymal complex, homologous chromosome. So this is talking about meiosis 1. So in meiosis 1 again we can find 4 phases: prophase 1, metaphase 1, anaphase 1 and telophase 1. So if we take the first one, A, occurs in prophase 1, and B, formation of synaptonymal complex. So what's this synaptonymal complex? The synaptonemal complex is a complex of proteins between the homologous chromosomes. So they form a complex, and that tight pairing of the homologous chromosomes is called the synapsis. So I will just draw the synaptonemal complex here. So we have one chromosome here and the homologous chromosome here, the next one. So they are, they form a complex here. So there is a this complex is called the synaptonemal complex. This tight pairing of these homologous chromosomes is called the synapsis. Now, in option B, it says formation of synaptonemal complex, again that occurs in prophase 1. Then C, pairs of homologous chromosomes arrange on metaphase plate. So that is in metaphase 1. So I will draw the cell in metaphase 1. So here pairs of homologous chromosomes arrange on metaphase plate. That is this plate. So we can see pairs of homologous chromosomes. These pairs arrange on metaphase plate. So option C is in metaphase 1. Then D, crossing over of chromatids. Crossing over is the exchange of parts of these chromatids. So in crossing over, parts of chromatids break, exchange and rejoin at corresponding points. So here, if I take this same set of homologous chromosomes, in crossing over, they exchange parts of chromatids. You can see the parts of the chromosomes that have been exchanged with each other. So that is the crossing over. Crossing over occurs in prophase 1. But after the formation of synaptonemal complex, first synaptonemal complex has to be formed and then the crossing over occurs. So B should come before D. And in all these answers you can see B before D. And we have 3 options in prophase 1 and only 1 in metaphase 1, that is C. So now we know that C is the last event out of these 4 that is in metaphase 1. So C should be the last one in the answer. So here from that knowledge we can reject 3 of these answers. We can reject 2, 5 and 3 because C should be the last one. Right, now in 1 and 4, C is the last one. Now we have to decide which one comes first. Now here C is the last one in both the answers and B is before D. Then we have to think about A. A is centrosomes move towards opposite poles forming spindle. Now the movement of centrosomes start at some point in prophase 1. It is not clear to me which one begins first, the synaptonymal complex formation or the movement of centrosomes. But we know that the movement of centrosomes ends towards the metaphase 1. So we can think that this should happen after the formation of crossing over, because the movement starts at the beginning or in the middle of this prophase 1, but it ends towards the end of the prophase 1. So we can say synaptonymal complex formation occurs first and then the crossing over, and then after that centrosomes move towards opposite poles forming spindle. So the answer should be B first one, D second one, A third one, and C should be the last one. So B, D, A, C is the answer number 4. Then question number 5. Which of the following statements regarding photosynthetic pigments is correct? First one, chlorophylls absorb yellow and blue light and reflect green light. Chlorophylls absorb blue and red light, not yellow light. So they absorb blue and red light and reflect green light. So option 1 is incorrect. Then option 2, chlorophyll B prevents the formation of reactive oxidative molecules. That is not a function of chlorophyll B, that's a function of carotenoids. So 2 is incorrect. Then number 3, chlorophylls and carotenoids are located on the membrane system of thylakoids. Yes, these photosynthetic pigments are located on those membrane systems of thylakoid membranes. So 3 is correct. Number 4, carotenoids and chlorophyll A absorb light corresponding to the same wavelengths. To see that, we need to take the absorption spectrum. So here we have the absorption spectrum. In that, we need to check for carotenoids and chlorophyll A. Chlorophyll A is in blue here, and carotenoids in purple here. So we can see they absorb light corresponding to different wavelengths. Chlorophyll A absorbs light somewhere in between 400 and 450 nanometers, as well as 700 nanometers, and carotenoids absorb wavelengths around 500 nanometers. So they are different. So this option 4 is incorrect. It says carotenoids and chlorophyll A absorb light corresponding to the same wavelength. So it is incorrect because they absorb light corresponding to different wavelengths. So option 4 is incorrect. Then 5, according to action spectrum, chlorophyll B is more effective for blue and red light. So for that we will take the action spectrum. So in the action spectrum, here the second one is the action spectrum. In the action spectrum, y-axis is the rate of photosynthesis and the x-axis is the wavelength. So it gives the rate of photosynthesis at different wavelengths. So we can see the rate of photosynthesis is higher, around 450 nanometers, between 400 to 500 nanometers, and around 700 nanometers. So it gives the rate of photosynthesis. But here option 5 says, according to action spectrum, chlorophyll B is more effective for blue and red light. These regions are blue and red, that is correct, but it doesn't, the action spectrum alone does not give any information about chlorophyll B. If we want to compare these wavelengths with the pigment, we have to overlap or compare both the action spectrum and the absorption spectrum. In the absorption spectrum, the red line is the chlorophyll B. We can say its amount of light absorbed is high at near 500 and near 700 nanometers. They are the blue and red wavelengths. Chlorophyll B is more effective for blue and red light. Chlorophyll B is more effective for blue and red light. That is correct, but we can read that information from the absorption spectrum, not from the action spectrum. So option 5 is incorrect. The correct answer for this question is number 3. Chlorophylls and carotenoids are located on the membrane system of thylakoids.